Here are 4 high-yield USMLE-style questions focused on cilia structure, motility, and associated disorders. Question 1. A 6-year-old boy presents with chronic respiratory infections and a history of situs inversus. Genetic testing reveals a mutation in the dynein arm of cilia. Which of the following is most likely disrupted in this patient's condition? A. Movement of motile cilia. B. Function of non-motile cilia and signal transduction. C. Cell division and mitosis. D. Cell membrane fluidity. E. Phagocytosis and respiratory epithelium. Answer: movement of motile cilia. Explanation: the dynein arm is crucial for the movement of motile cilia. It links the microtubule doublets in the axoneme and causes bending of the cilium. Mutations in dynein lead to defective ciliary movement, which can result in Cartaginer syndrome, characterized by chronic respiratory infections, situs inversus, and other defects due to impaired ciliary motility. Question 2. A patient with polycystic kidney disease, PKD, is found to have mutations in the genes encoding non-motile cilia. Which of the following is the primary function of these cilia? A. Chemical signal sensing and signal transduction. B. ATP production for cellular energy. C. Intracellular protein synthesis. D. Cellular adhesion to the extracellular matrix. E. Production of extracellular matrix proteins. Answer: A. Chemical signal sensing and signal transduction. Explanation: non-motile primary cilia act as cellular sensors involved in signal transduction. They play a role in regulating cell growth, differentiation, and response to chemical signals. Dysgenesis or dysfunction of these cilia is associated with diseases like polycystic kidney disease and retinal degeneration. Question 3. A researcher is studying the structural components of motile cilia in human epithelial cells. He observes that the cilia have a 9+2 arrangement of microtubules. What is the primary function of these motile cilia? A. Sensing extracellular signals for cellular response. B. Propelling the cell through extracellular fluid. C. Generating force to move fluid across the cell surface. D. Maintaining cell structure. E. Synthesis of cellular proteins. Answer: C. Generating force to move fluid across the cell surface. Explanation: motile cilia have a 9+2 arrangement of microtubules, 9 doublets around a central pair of microtubules. They generate movement by sliding of microtubules mediated by the dynein arms, allowing the cilia to beat and move fluid across the surface of cells. This is critical in the respiratory tract, fallopian tubes, and other structures. Question 4. Which of the following findings is most likely to be associated with defective ciliary function in a patient with retinal degeneration and mitral valve prolapse? A. Increased susceptibility to bacterial infections. B. Abnormalities in signal transduction pathways. C. Impaired function of microvilli. D. Disruption of ribosomal function. E. Defective glycolysis and ATP production. Answer: B. Abnormalities in signal transduction pathways. Defective non-motile cilia, which function as signal sensors, can disrupt signal transduction pathways, leading to various diseases such as polycystic kidney disease, retinal degeneration, and mitral valve prolapse. These cilia are involved in cell growth regulation and response to chemical signals, and defects can lead to developmental and degenerative disorders. These questions cover